Recording in progress.
Yes. Hola, buenas tardes. Gracias por estar aquí con nosotros, acompañarnos de manera presencial y por Zoom. Realmente es un gusto presentarles a la doctora Alexandra Maertens de la doctora Maertens, su grado de licenciatura en el colegio Excelsior en Albany, Nueva York. Después hizo un certificado en Data Mining en la Universidad de California en San Diego, para después unirse al laboratorio del doctor Thomas Hartung, también en el Hopkins, en la Escuela de Medicina Pública, en la División de Toxicología, donde realizó continuó por 2 años más para después ser contratada en el Johns Hopkins, también en la Escuela de Medicina Pública, en el clínico de laboratorio, donde a partir del 2018 fue investigadora y en el Johns Hopkins. Pertenece a uno de los principales es manejo de riesgos medioambientales como toxicóloga, al cual pertenece desde el 2010. También está asociada al Fondo para el uso de la bioinformática y la innovación en cáncer, donde participa activamente desde el 2018. Participa activamente en actividades de docencia en la Escuela Pública de Bloomberg del Hopkins, en el Departamento de Informática de la Universidad Ramsey y en el Instituto Tecnológico de Pensilvania. La doctora Maertens ha publicado varios artículos muy relevantes en revistas de alto impacto, donde se enfoca a un análisis diferente, con un punto de vista diferente del data mining, de que nos va a platicar un poco de eso el día de hoy. Y también ha participado activamente en hacer extensiva la ciencia a otras comunidades, participa en diferentes podcasts para gente joven en investigación, como jurado en diferentes premios para estudiantes que se dedican a la investigación. También ha sido muy exitosa en obtener funding para sus proyectos, tiene varios grants vigentes y pues no me queda más que darle las gracias a Alexandra por haber aceptado su participación acá en el programa, por la clase de ayer y por el seminario de hoy. Thank you very much, Alexandra. Adelante.
I hope that was good, and my microphone is on, so we are going well. All right, so I'm here to talk to you about environmental health. I do work in bioinformatics. I spend a lot of time with genes, but fundamentally I'm a toxicologist and I work at the School of Public Health. We try to focus on looking at the environmental aspect of it. But one thing I've learned when I work with oncologists is that we have done amazing things for tertiary disease prevention when it comes to cancer. We can sample your tumor, we can figure out its unique weakness, we can dose you with it in ways that are far less toxic than it used to be. This is true not just of cancer, we have better drugs now for type 2 diabetes, all sorts of pharmaceutical interventions that are uniquely targeted. But we've made very little progress on slowing even subclinical disease down. If you have metabolic syndrome, it's still, we can push lifestyle interventions on you, but there's not much we can do. And if you go even back further to the environmental origins of these diseases, we've made very little progress. The omics data sets have contributed very little to this. So I'm here to talk about why that is and what we can do to fix that. So before I dive into genes, though, I'm just going to talk a little bit about chemicals and why they're important. So I know you may not think a lot about this, but there are a lot of chemicals that you're exposed to every day. Some of them are benign, many of them are not. All of us are exposed to a certain level of arsenic. It's in the Earth's crust, lead, it's inevitable. You're going to be exposed to it, air pollution, man-made pollutants, etc. But can we really tie those chemical exposures to DALY or disability adjusted life years burden? The answer is no, we really can't do that. Aside from air pollution, it is very difficult to connect chemical exposures to adverse outcomes in people. So air pollution counts up about 70% of what we know. And I think it's a little bit sobering to think that gasoline would never be approved under current regulatory regimes. It's a known carcinogen. It's a terrible substance, but once we let it in, we can't get it out. The things that people think about, like long-term chemical exposures, actually account for very little of what we know about. There are occupational exposures that we do know are quite bad, as well as acute poisonings, but fundamentally there is a real lack of knowledge in this space. So we do not have a lot of quantified exposure-response relationships that are supported by very strong evidence. There are many chemicals where their health outcomes are simply completely unknown. Large-scale exposure data is insufficient. Most people are kind of surprised, but we actually do not have a catalog of every chemical you are exposed to. We don't even have the analytical tools to figure that out. And there is a considerable knowledge gap in terms of how this impacts the population. So the health impacts also from exposure to polluted sites are very difficult. There are about 56 million people worldwide that have very locally specific health impacts that we can't really think about. A lot of these are located in third world countries. For example, e-waste is a significant problem now in Africa. There are air pollution problems throughout the world that are quite local and quite specific. There are industrial effluvians for solvents, things like that, that are going to have a very high local impact, but not a global impact. Those are extremely hard for us to quantify. And a lot of times we have had big data, but big data of the wrong kind. So a good example of this is bisphenol A. It's considered an endocrine disruptor. It's the kind of chemical that scares a lot of people. It's been subjected to multiple guideline studies. There are 10,800 abstracts in PubMed for bisphenol A. I have personally contributed 2 of them, so sorry about that. But this is a lot of data for one chemical. If you look at the main database, it says that it affects 2,000 genes. There's 79 studies with laboratory studies, and yet, with all of this data, the last FDA review found that none of these could be used for risk assessment because the studies lacked adequate statistical power. So this is a ubiquitous chemical. If you are exposed to plastics, you're exposed to some of it. We've done a lot of data. Do we have competent regulations on it? We still do not. And another kind of class of chemicals to think about are flame retardants. So flame retardant exposures are ubiquitous. If you buy a couch in the United States, about half of the weight is a flame retardant because they're very heavy. Their fluorines are quite considerable. Children are exposed to very, very high amounts of them because they tend to eat, if you've ever had a child, that anything they see off the floor, they pick it up, they put it in their mouth. So they have a much higher exposure than adults do. There are 500 discrete chemicals that are classified as flame retardants. So because there were so many of them, they figured that the only way that they could analyze them by class, you probably heard of some of them, organophosphates, polybrominated esters, they're very commonly found. So how do we kind of figure out how many chemicals we are exposed to? So even if you take that small, narrow class of 500 chemicals, and you look at the metabolites, most of those chemicals have at least 10 metabolites when you use the normal programs that your average, the toxicologists tend to use. We used 2 of them. One was conservative, one was not conservative. They overlapped a little bit, but fundamentally, there is a combinatorial explosion of possible chemicals once that chemical hits the metabolic system. And in many cases, a lot of these classes, there's different classes, they will share the same metabolite. So if you are taking a blood sample and you look for that metabolite, you actually cannot trace it back to the exposure because it could be either of them. Most of these metabolites are not in the databases used to identify biomarker samples. So they will just disappear if you're doing a mass spec study with a blood sample. And this had amazed me, but you should always, understand that cytochrome P450s are amazing enzymes. 7 flame retardants were metabolized into other flame retardants. So it is very difficult in this chemical soup that we're exposed to figure out what people are exposed to, what metabolites they are exposed to, even if you had perfect mass spec technology for their blood. So the sheer numbers make it difficult. So when I started as a toxicologist, my first task was mapping the human toxome project. They said, well, the number of chemicals is quite large, but the number of possible responses in a cell, that is finite. We can map that. And we thought we could. What we did discover was that the cells that we were using to map these were not terribly reliable. We were using MCF7 cells. And after years and years of work, one thing that we discovered is that they're genetically unstable and they will give you very irreproducible science. So that was a frustration. They then later used bioinformatics and some cancer data to prove this in excruciating bioinformatics detail. They are not the same cells. They are not the same biology as a normal human. And so this taught me a useful lesson. One was that even if we think we have the tools to sort of translate, how a cell responds to a chemical challenge, we actually have only a very limited peek of what actual human biology can do. But as all bioinformaticians will do, when I discovered that there was an enormous amount of data on cancer, I decided I will study cancer. So I grabbed all this data and I focused on something that I had noticed when I was studying these molecular responses to estrogen in a cell. That if you looked at all these genes that we knew were important to cancer, and we just sort of took all these genes together and asked whether they were significantly associated with survival or Gleason score or whatever, that there were a ton of genes that were just consistently overlooked in the literature. They got no attention, they had less than 50 PubMed IDs, they didn't appear on pathway databases, they were for all practical purposes invisible or not studied at all. And the gene annotations were missing systematically a lot of biology. RNA splicing tended to disappear. We had more primate-specific genes in these genes that were poorly studied. So I realized that there was just this wealth of biology that was also sort of disappearing if you use the normal approaches. And I also discovered, however, that if you had a very, very large data set, we could start to see some of the challenges of linking genes and exposures to environmental outcomes. And I'll give you just kind of one sample of the work that I recently did. So the MUTYH gene is a key base repair enzyme. If you are homozygous for a dysfunctional version of this, it's 100% penetrance. You will get colon cancer usually by the age of 50. So this is a very clear genetic mechanism for cancer. The problem was for heterozygosity. There had been conflicting epidemiological studies about cancer susceptibility, and it remained very difficult because, for one thing, you tend to have very small sample sizes. Heterozygosity in the European population is about 4%, so it's not small compared to some genetic SNPs, but it's not large. If you want to get several thousand patients, that's going to be difficult. And you always have confounding exposures, so it's a big challenge. So one of the things we did is we realized if you are willing to cooperate with industry, you can get a very large data set to answer exactly this problem. We had 354,000 patients where their tumors were sequenced. And thanks to a nice algorithm that allowed us to infer the germline status from the somatic status, we could figure out whether there was a loss of heterozygosity from a tumor sample without having to take normal matched tissue. For a lot of cancers, that is very, very difficult. For some cancers, it's just too expensive. So these sorts of numbers would have really been impossible if we hadn't had the algorithm to figure this out, as well as a really large data set that would have been difficult to get with any other means. And when we looked at this broad data set and all the cancer types, we did find that yes, MUTYH heterozygotes did show a very slight increased odds for many cancers. And they also had the distinct cosmic mutation pattern for base excision repair knockout. So we knew that this was likely causal rather than just an accident passenger mutation. But this would have been completely invisible if we had had even 10% fewer cancers. We really had to have this very large sample size in order to see this. The other thing we saw was that this was not true for all cancers. It had been debated in the literature for breast cancer, prostate cancer. We were kind of, we had almost 10,000 breast cancer patients in this data set. There was only one person with heterozygosity, so it turns out that the answer is almost certainly no. It does not increase hormonally driven cancers. They were either insignificant or just barely above background. So I think that there is a good news thing here. You can figure these things out, but you do need very large numbers. And there have been a lot of instances where people have used very similar approaches to start thinking a little bit more granularly about cancer causation. So we used to think that skin cancer was just sort of a function of how much melanin you have. People who look like me have very high rates. People who are a little bit darker have low rates just because they have better protection from UV. But it actually turns out if you look molecularly, they're getting skin cancer from different mechanisms. So they compared a Singapore population and a UK population. And in UK, you can see that there is a very distinct cluster that's in red of UV-mediated DNA damage. In the Singapore population, it was a little bit more dispersed. And what to me, it's very horrifying if you're as pale as I am, but you can see in the 1 millimeter skin population with the somatic mutations, almost all of the cells had some vast range of mutations, whereas in Singapore, it was really driven by notch mutations. So it is not just a linear function of your UV exposure. In point of fact, the people in Singapore were getting skin cancer, but it was a different mechanism of skin cancer. And I want to think a little bit then about how we talk about environmental diseases. The Bradford Hill criteria, which is how epidemiologists decide is there causation for an environmental exposure and disease, was sort of established studying cigarette smoking. But deep buried in that article, they said, you know what? Maybe atmospheric pollution could be a contributor to smoking. And then they said, no. They sort of put this kind of balloon out, looked at the data and said, no, when you really crunch the data, it really does look like it's cigarette smoking. They had a very large study on UK doctors. If you can imagine an era where doctors were smoking, it just says how much we've changed. So it turns out that if they had had more data and higher power, they would have been able to see that if you take out any lung from anybody who's old enough, there will be a lot of driver mutations, even in non-cancerous tissues. Your lungs are just exposed to a lot of stuff. They are going to get banged up. Their DNA will be mutated. However, if you expose to air pollution, you see these non-mutagenic mechanisms, for example, altered methylation in the EGFR driver, et cetera, that likely account for this huge increase we've seen in lung cancer and even in people who have never smoked. So again, if you have a large enough data set, if you have the omics technologies, you can start to kind of connect these exposures to outcomes in a better way. But that's cancer. In a lot of ways, this is kind of, to me, bringing environmental health back to the future. The very first carcinogen was actually discovered in 1775, and it was because Percival Potts noticed that there was a very, very rare disease, scrotal cancer, that only appeared in chimney sweeps, and it was connected to the soot. Now, different people dealt with this knowledge. In Germany, they just said, hey, let's make everybody wear pants, protective clothing. They knocked scrotal cancer out. They did not do that in other localities. So whether people actually translate this information into protective measures, it's a different thing. But traditionally, this is actually how we learned about carcinogenicity. So for example, we observed people in the dye industry had high rates of bladder cancer, people in the exposed to vinyl chloride, high rates of liver cancer. Those are very rare cancers. And so when you see them in an occupational scenario, you know what to look for. But we're not really doing that anymore. Occupational health has not been something that is emphasized a lot. We do have to kind of think about what occupational health is going to look like for the 21st century. Biomarker driven has to be for both exposure and disease mechanism, but critically, it has to capture casual and non-unionized industries. A lot of the highest exposures now are not happening in factories because we figured out how to fix a lot of those problems, but they are happening for agricultural workers, for cleaning staffs. They're exposed to very high levels of solvents. Nail salons have a surprisingly high level of exposure. This will be a very difficult balance between privacy and highly granular genomic and exposomic data. So think about everybody who donated their tumor to TCGA. We can now figure out their germline DNA. We've made a lot of advances that way. So data that seems private or doesn't seem that important. And I will say, if you've worked on an exposome project where you're just looking at blood samples, you get a pretty good record of what people are up to on Friday nights if you look at their blood. And it can make some populations very reluctant to share in this. So this will be a challenge in order to distribute this fairly and equally to everybody. And I also kind of want to point to one of the areas that I have worked on for a while, which is sort of trying to figure out developmental toxicity. And I also worked for a while in neurodevelopmental toxicity. Traditional toxicology, where we just dosed rats and we watched them get a little sick, it did not do a lot to change the risk factors for any of those diseases. We found out a lot about mechanisms. There was a lot of cool science done, but in terms of definitively tying chemical exposures to birth defects, we have a few wins, but mostly losses. On the other hand, if you mine electronic health records, a lot of things popped up that we probably would not have seen if we had not had very large data sets. So the anticholinergic drugs and a link to increased dementia has been repeated a couple of times. I think the debate is still open about diabetes drugs and paracetamol exposure. There's still a lot of debate about that. But the ability to sort of look at very large numbers and tie pharmaceutical exposures to outcomes has definitely changed the way we've thought about a lot of these things and the sort of numbers that you need to actually see it. So when you think about exposomics, and this is usually the challenge I run into when you talk to people who work in GWAS data, they always point out that the problem with the environment is too big, right? The space for GWAS is very, very large, but it is finite. The space for exposures seems to be infinite because you have to consider the disease history, the individual health status, everything they've been exposed to over time. So I don't want to sort of emphasize this because the problem is there really are no exposures that individually are smoking guns. If they were, we would probably have figured that out. What you do probably have is this accumulation over a lifetime of very small hits to areas where you are uniquely genetically vulnerable. And over time, that accumulates in diseases such as diabetes or Alzheimer's or Parkinson's. This will require a lot of new data generation. And I just want to take a brief moment to talk about a very unique type of data that we work on at the School of Public Health, organoid intelligence. So one of the things that has been a challenge has been neurodegenerative diseases and neurological diseases in general. We've gotten pretty good at growing some aspects of of brains in little petri dishes. And usually there's a lot of skepticism about this. People say, well, how good? So good that they can play Pong. So this was done by a local, somebody from Mexico City, it's Morelos Pantoja. You can actually it takes about, a couple of flasks full of mini brains. And if you tell them to play Pong and, you connect you have to kind of connect the electrodes into the microelectrode array, and then you have to do something for your output. It's not simple to do, but they can play Pong pretty well. They actually get really good at it in about 5 minutes, and you can actually see the energy demand of the brain spheres going down. And then when they miss and the ball goes off, you can see their energy go back up. So we have a pretty good granular readout, and we can dose them with alcohol and watch their playing get worse, etc. So I don't want to oversell this. We don't have a blood-brain barrier. We don't have the immune component. There are so many things that we are lacking in this model, but there are so many things that we can also capture. So we have used this. We've used the microphysiological system to kind of look at some organotypic effects for neurological diseases. We have used this to demonstrate a gene-environment interaction between a known autism gene, CHAD8, and chlorpyrifos. Chlorpyrifos is a pesticide that we have been trying to get banned for about the past 20 years. It's been associated in epidemiology data with ADHD or autism, but the rates were always very low, and it's very easy for industry to kind of argue, well, you're looking at this teeny tiny change, how do we know that's real, et cetera, because the mechanism wasn't there. We do think that we've kind of established that with this at least susceptibility factor, which is pretty rare, but it is kind of the first gene-environment interaction that we can point to for a neurological endpoint. But when you look at all the possible SNPs that are associated with autism, there are 1,000 SNPs just at the protein coding level, and there's another 700 epigenetic sites. So how can we use functional genomic predictions that we kind of use from precision medicine data sets to narrow the search space a little bit? We decided that for this first kind of proof of principle concept, we weren't going to focus on autism because we felt there was too many challenges in the diagnosis, as well as the differences in the way this data was gathered. And there wasn't really a lot of mechanistic information that we could apply to most of our studies. So we decided we would look at a slightly easier problem because at least most of the mechanisms were well established. So we have some collaborators that were working on obesity as a modifying impact of asthma. And they said something that kind of made me perk up. They said, we've always thought these diseases had a common genetic underpinning, but we've never really known that they did. So both asthma and type 2 diabetes are polygenic diseases. They consist of multiple small effect genes. And the comorbidity between asthma and type 2 diabetes has been very well established, but the nature of the causation has been unclear. So the excess weight of obesity can affect lung mechanism and function. It can also impair your ability to do physical activity. Physical activity can increase your exposure to indoor air pollutants. Indoor air pollutants are worse than outdoor pollutants usually, so that can exacerbate asthma. Or asthma can just decrease your likelihood for physical activity. So we don't know which direction the causal arrows go. The polygenic risk scores for both of these has been pretty limited. I say they're limited. The people who do GWAS are like, yay, we predicted 70%, and they think that that's sort of a huge success. But usually the 70%, for example, for asthma is for childhood asthma, which has a much stronger inherited component. And I think that for type 2 diabetes, it's been a little bit more of a mixed bag. It depends how you define it, which population you looked at. But they too tend to get to this point what they call the missing hereditability, the part that they can't explain for genes. I'm always the person who's like, well, maybe that's the environment. Maybe we should think about that. We do think that the underlying genetic factors and epigenetic factors common to both are likely overlapping inflammatory and hormonal factors. So both of these diseases are known to have an environmental component, but the causative factors are very few, and they're also very poorly quantified. But we do know one thing without a doubt, and that's they are both increasing. Asthma rates and type 2 diabetes have just done nothing but go up in the past 20 years. And it's a small achievement that it used to be that most asthma cases that we saw in adults were driven by occupational exposures. Now we think that most asthma in adults is driven by being overweight. So, it's an important public health question. The known gene environment interactions in this case are actually, and by environment here, I'm meaning exposures to chemicals, not just the broader environment, are even fewer. So we pulled all of the GWAS data from catalogs for 3 endpoints: asthma, atopic asthma, and asthma exacerbation measurement. These are actually kind of different questions. Atopic asthma is a little bit different from asthma. Asthma exacerbation measurement just happened to be there, so we kind of tossed it in. We decided that we would take a broad view and just use all the genes. And when you do this, you can kind of see that chromosome 6, which has a very large number of immune genes, is just sort of popping right out. There's also a lot at 17, but you can see that this is what you normally see with your average polygenic disease. There's a bunch of hits that are very clearly, correlated, that are very high. Those are all the HLA genes. Everything else is a little bit more, statistically speaking, difficult to establish. This is not atypical for polygenic disease, but most of the SNPs that are protein in near protein coding area are either synonymous or intronic variants. So in other words, this is not a disease of proteins that don't function. This is a disease of proteins that are being regulated incorrectly. So if you're looking for the smoking gun enzyme, it's not there. It really is a regulatory thing. And we think that these regulatory things will sort of be more obvious if we think about what sort of stresses the regulation is under. I want to say that a lot of our signal, and again, this is common in any GWAS study, is located in non-coding regions or non-coding RNA. We spent a lot of time trying to figure out how we could include that. And we just decided to let it disappear because there was not enough information about what those do from a functional perspective or a regulatory perspective. So there's a huge part of our signal that we just had to kind of let go because there just wasn't much we could do with that. And you would think asthma, well-studied disease, when you just pulled out the top 15 genes and asked a simple question, how many PubMed hits? 11 out of 15 had fewer than 200. And 11 of the top hits were protein coding, 3 were non-coding. 1 had a pseudogene that was surprisingly robust. And you can see again that most of these genes that the hit is in a regulatory region for the lower scoring ones. When you looked at diabetes, you found the same thing. You found a handful of hits that were very strongly expressed. Fundamentally would not see a different distribution. So in terms of how the GWAS data looks, these are both telling us a similar story. You have a lot of hits in the intronic or the regulatory region, very few hits that are going to be a missense protein, and it's distributed in a lot of ways. So the first basic question we asked was, do the genes overlap at the protein coding level? And the answer is yes, that is more than you would get by chance. But we can't really be sure that that's telling us anything because a lot of these studies date from before the era of the UK Biobank. So a lot of these were targeted studies. So we don't know that this isn't just publication bias. People are looking at genes that they know are important. So the next question we asked, which any bioinformatician is going to ask, is what if you look at the pathway level? And the answer is at the pathway level, at least at the top pathways, they are different diseases, and different diseases in exactly the way you would expect. The top pathway for type 2 diabetes is insulin signaling, and it's the JAK-STAT pathway for asthma. There were only, in the entire data set, 2 pathways that overlapped. One was the PDGF pathway, and the other one was the gonadotropic hormone pathway. However, they did not overlap with the same genes. So in fact, in the gonadotropin, there was only 1 gene, but that 1 gene was an important gene. It was IRS1. And this was kind of our first clue that we needed to not just think of the numbers of genes, but what those genes were doing in that pathway. Although we did not realize it at that point. So, and I'm sorry for the bad data visualization. I literally just got this like last night. But this messy looking cluster of genes is what happens when you ask an algorithm to say, are these genes connected by known protein-protein interactions? And the answer is they're almost all connected by known protein-protein interactions. So we did this many ways. We did this just with the diabetes genes. The answer is yes. Just with the asthma genes, the answer is yes. So GWAS genes are not distributed just randomly across the genome. There are a handful of network level things that are going to lead to diabetes. And if you look at it this way, it is a much more consistent picture than if you just ask at the pathway level. And then when you looked at all of them, they also overlapped. And the red genes that you can kind of see sort of peeking out at this messy little hairball, those are the genes, the 124, that overlapped in both data sets. And I think this is really critical to understanding how and why these diseases might be comorbid. These genes did not cluster on any pathway. These genes were not barely significant for protein-protein interactions, so probably not really. They did not have any pathway annotations. You can see they're just sprinkled out in this little hairball. So insofar as there is an overlap, it's not that they have the same pathway in common, is that they have the handful of transcription factors that are quite critical for multiple pathways and are likely combining these things. And so if you think of these diseases as diseases of networks, then they really are very, very similar diseases. And the pathways will give you a more distorted view of that because it thinks of those pathways in isolation. So asthma and diabetes, do they overlap? At the gene level, yes. But most polygenic diseases are probably going to have some level of overlap. At the pathway level, kind of. But this may obscure a lot of connections between pathways that are not actually discrete, or genes such as IRS1 that are really critical. But at the network level, yes, both diabetes and asthma are highly significant and rich for known protein interactions, as well as in the combined network. And fundamentally, the mean pathway length between diabetes genes and asthma genes is about the same as between asthma genes and diabetes genes. In other words, they have the same distance from each other in this network. And it is very much smaller than you would get by chance from a network drawn from random genes. However, GWAS genes are not random genes. So in order to truly answer this question, you can't just say, well, look, it's more than random genes. You have to build a network that sort of resembles the same distribution in terms of transcription factors. So that is what we're working on now. And I think we're probably going to find that, yes, it's more than by chance, but that polygenic diseases just have this more often than you would expect by chance. So it really is a handful of transcription factors that do a lot of things. So the next thing we thought was, okay, we have to tie this into the environment somehow. And as toxicologists, we thought the obvious thing to do was to look at what mediates your relationship with all the chemicals you're exposed to. So we looked at ADME genes. So there are about 300 genes. This is a list that was created because the pharmaceutical companies really, really needed to know which genes were being up and down in response to exposure. So this consists of enzymes, cytochrome P450, glutathione, signaling genes that are involved in xenobiotic metabolism, et cetera. And these we thought would be an obvious place to look because these genes tend to be very highly polymorphic in human populations. There is a lot of variance in your xenobiotic enzymes because most of us have been exposed to a lot of different chemical challenges and diet, et cetera, over time. So we thought this was an obvious place to sort of say, aha, we found it. Here's where the environment is impacting your susceptibility to disease. That turned out not to be true. This was what we thought was going to be the story, but there are very few ADME genes that are implicated in either diabetes or asthma. There's one cytochrome P450, glutathione transferase, but a list of 300 genes that is not significant. There are a few transporters, but they didn't really overlap. The only overlap we found between the two diseases was ARNT, the aryl hydrocarbon nuclear translocase. This is a very, very important gene for aryl hydrocarbon mediated response. So this is possibly a place where the environment is impacting both. But we didn't find a lot of evidence for that when we looked at the epigenome studies. So again, when you kind of think about these diseases, we thought this would be an obvious place to look. It turned out not to be. So we had to go back to the drawing board and we thought a little bit about, well, where else does the environment impact your genome? And I remembered something that I was really surprised I did not see in the diabetes genes. I did not see lipid transport. It did, it was way, way down there. It was barely significant. I had a little bit, if I'd made my cutoff slightly less stringent, I would have seen it, but I didn't see it in the reactome pathways, things like that. And I thought, well, that's really strange. But then when I pulled the reactome pathway for cholesterol, it was a list of enzymes in this sort of linear pathway. And of course, diabetes is not one enzyme or two enzymes in this pathway. It tends to be, again, transporters or signaling molecules. So I thought, okay, we need to do a slightly different method. So if some of you are familiar with string, you may not know that there is a small molecule version of it that just connects genes to any small molecule you want. So we started with cholesterol because it seemed to be a very obvious place to kind of prove that, yes, there were some significant things. So we built a high confidence map of all the genes that were known to have physical interactions with cholesterol. And this gave us what we thought was a much better mix. It's a combination of transporters, enzymes, as well as transcription factors like SREBF1 that are critical for cholesterol homeostasis. So when we looked at this, the network impacts in terms of the GWAS were pretty few. There were 5 for diabetes and there were 3 for asthma, so that's not a whole lot. but they were very important. The one place that they overlapped was ApoE. I'm sure most of you have heard of that gene. You've heard it in the context of Alzheimer's. That's how I heard about it. What I did not know is that this is the one SNP that is, or this gene is the one gene where SNPs consistently impact mortality, even if it doesn't, if you're not including Alzheimer's. In other words, having a dysfunctional version of this gene is bad news, even if it's not bad news in the way we normally think about it. And in this case, I did check the SNP that was in common between the two of them was actually in the regulatory region. It wasn't the epsilon that we're familiar with. Asthma also included LRP1. This was the highest degree in our protein-protein interaction network. So this was the central, one of the most central ones you could find. It had what's called a very low human gene damage index. So that is just an index of how intolerant a gene is to protein coding mutations. This gene is very intolerant, which means that there are very few reported protein coding mutations in it. And this indicates, this is kind of one indication that having a SNP that impacts the regulatory area is going to be a problem. And then what we really wanted to capture was when we did text mining, it indicated that it covered a very diverse biological space. So we did a very kind of primitive version of asking what's the semantic space that this gene was involved in. We just did a metric on OMIN to try to figure out how many processes it was involved in. We're currently trying to refine this, improve this a bit better, because we think this is kind of the key to thinking about how these genes impact the molecular networks, is understanding whether these genes are one enzyme doing one thing or a signaling molecule that is coordinating between, say, energy metabolism and inflammation and apoptosis, because those genes are likely the ones where you're going to see the most impact. When we looked at the epigenome-wide studies, we did also see extensive alterations of this molecular network. So diabetes, you had 15 of 88 hits, asthma, 7 of 88 hits. And if you added air pollution to this, you got even more. And you won't be surprised that air pollution is targeting some of the more important genes. So it hits LRP1 and STRABF. And I know when I talked about this, people are like, well, air pollution and diabetes, there's plenty of people with both those conditions and exposed to a lot of air pollution. So your potential to get multiple hits and multiple important hits on this pathway is certainly there, depending both on the luck that you were born with, the diseases that you develop, and the exposures that you have. We are currently repeating this for 20 different micronutrients, because one of the things we thought, looking at cholesterol, we thought we made our case that cholesterol was pretty important to both these diseases, but then our question was, what's the null hypothesis here? So we thought, let's do a literature review and see if there's micronutrients that are definitively not important for diabetes or type 2 diabetes. There were no micronutrients where everybody agreed that was not the case. So we gave up on that. What we did do was decide that we could kind of look and see if there was some discriminatory power. So we did that with vitamin D. Again, we had significant hits. They were different. There was very little overlap between type 2 diabetes, but the GWAS hit actually 8% of the entire network. Folate had very few hits, but that one hit was mTOR. That again shows you the importance of thinking about not just the number of hits on a pathway, but how important that gene is. So our hope with this is that we can rank the potential gene-environment interactions, not at a pathway level, but at a network impact level. So we can really think about combining the scores for degree, the gene damage impact, and the semantic space that we indicate with the annotations and text mining. And so for us, this is sort of the first step in dimensionality reduction for prioritizing the gene and environment and interactions, not just that we can say, yes, asthma and diabetes do have some common confounders, but there are common underlying genetic predisposition, but this predisposition is probably going to be modified by your lifetime exposure to cholesterol, as well as your lifetime exposure to air pollution. And here's a hypothesis we can test for that. So we can kind of put this at a more mechanistic and hopefully a more quantified level. But I want to kind of go back to thinking now about plastics. As a toxicologist, it kind of always has to go back to the environment. So I want to point out that half of all plastics that have been ever produced since that have ever been made have been produced since the year 2000, and it's only predicted to go up. Most single-use plastics are used for a day, but they last exponentially longer. And this is going to continue to be a problem that will haunt not just the young people here, but your offspring. They don't go away. It's estimated that Americans drink 42.6 billion 1-liter bottles of water a year. Globally, it is 200 billion plastic water bottles. And each plastic water bottle can take up to 450 years to degrade in a marine environment or a landfill if there's no photodegradation. So all of that every year, stacking up, will not disappear. If you've had any experience with dynamic systems modeling, you can just think that's going to increase over time in a terrible way. How did this happen? Well, in a lot of places, bottled water is perceived as safer. People do not trust the science when the agencies are entrusted to keep the tap water safe. I live in DC. I know how to check and call my local EPA and look, I know the tap water is safe. I cannot, even though I'm a toxicologist, convince my friends to drink it. They're like, no, bottled water is better. But in many places, the very poor infrastructure means that people are right to mistrust their public water. This is a tragedy. We can fix this. This is a simple engineering problem, but we have let this happen. So, a lot of times people focus on the toxicity of the additives such as BPA, but that is not important. What is important is all these plastic water bottles. How do we fix this? Okay, we can recycle, but this will create plastics with potential contaminants. And there was just a study that came out that said that a lot of food plastic contaminants has flame retardants in them because they're commonly added to polymers. We can incinerate, but that will create combustion byproducts, many of which will be toxic, and the exposures will likely be focused in disadvantaged communities. We can switch to glass, but that will create increased greenhouse gases because glass can be substantially higher in terms of its greenhouse gas impact. We can use new technologies. We can create bio-based plastic that rapidly decompose in industrial systems, but that will require research and development. It will require rapid-acting toxicology that can conserve safety and an investment in infrastructure. And I can tell you, as somebody who deals a lot with both the EPA and the public, that telling people, don't worry, we've tested it, it's safe, that is a hard sell right now. It was a hard sell in the US for vaccines. And if that continues to be, we will not be able to make the sort of technological changes that we need, because all of these choices are going to require balancing trade points that are very important for human health. So we need to think about that so it is done fairly and so it is done equitably. So thinking about gene-environment interactions and quantifying the environmental impact, it is, if you'll excuse the pun, not just academic. And I think that the important thing is that we all know, I think if you're a scientist, that we have to have a green economy. We have to change the way things have been. But avoiding global warming will require a massive reduction in red meat, dairy, all the things we love. We will have to re-engineer our food supply in pretty drastic ways. But this can have unforeseen consequences that traditional health approaches will not easily identify. For those of you whose chemistry is not recent, that's a cis fatty acid and that's a trans fatty acid. One of them is fine. One of them will lead to heart attacks if you eat enough of it over time. And we found this out the hard way by manufacturing margarine with large amounts of trans fatty acids and eating it. Sometimes we recommended people to eat it to avoid the health effects of cholesterol. And we found over 20, 30 years, only with epidemiology studies, that this was a very bad choice. So when we think about all the changes we will have to make, we have to come up with a better way to more quickly identify these dangers, figure out who is susceptible to them, and figure out how to fix them. So with that, that is my email. There's some sort of place that I think it used to be called just Twitter. You can follow Kat on that. I'm on Blue Sky. I have made the change, but you can follow me there. Kat has a newsletter if you want to see super cool things like brains playing Pong in a dish. You can sign up. I want to thank Dr. Fenicillier, Thomas Hartung, the entire Kat team, but most importantly, all that data that you saw was not done by me, it's done by my students. Brianne Kincaid, Jason Laird, Daniel Kegbu, Alan Kim. And if you thought this was cool and you're interested in toxicology, I wrote a book. And with that, I will take any questions. Or we can just enjoy feedback.
About the study that you showed for diabetes and ApoE1, ApoE,
Mhm.
Diabetes is related because most of diabetes persons die from atherosclerosis.
Yeah.
Is there a statistical correlation or correction for those kind of bias?
I mean, they corrected the bias as far as they can. Probably you never truly get out the confounding factors. I will say that apoE at subtypes probably starts a fire that is going to burn some organ out, and it's a question of luck and time which one it is. In terms of the mortality statistics, they've looked at the mortality statistics in a lot of places where the impact of Alzheimer's is blunted because people tend not to live that long, and it was still associated with non-communicable diseases having lesser impact. So I think that, and again, I'm not a specialist in epidemiology, but from what I understand, the agreement that having a problem in ApoE is bad is pretty consistent. It stands out more in Alzheimer's than others. But if you look in the UK Biobank and just all the parameters altered that it's correlated with, it's a lot. So at a simple level, the phenotypic consequence is pretty significant. The one thing I will say is that in terms of the gene damage index, it actually doesn't rate very high. It's kind of one of the middling genes, if I remember correctly. So there's some genes that are very intolerant, some that are super tolerant, and then there's the middle. It's in the middle. So it is probably one of those genes that was not under a lot of selective pressure because its effects were not felt until you were in your 60s or your 70s. Sure.
For the study for the organoids, there is a little bit of a problem with the nomenclature. So I'm not quite sure if organoids is meant for repair from stem cells or is spheroids.
They're all prepared from human stem cells, and the advantage of that is that we can capture human genetic diversity. So for the chlorpyrifos study that they did, they have a bunch of patients that are known to have the CHAD8 mutation that is linked quite highly with pretty severe autism. So these are not the borderline cases, but this is the unambiguous autism. So they can take skin cells from entire family, afflicted and non-afflicted. So they can minimize they can do a very controlled study on that particular SNP. So then when they grow the organoids from that, they can then, basically bomb them with chlorpyrifos and its metabolite and see the differences in neural outgrowth and things like that. So they call them mini brains. They are not mini brains. They're clusters, clusters of neurons and glia living happily together, for about 20 days. But I do think that the fact that we can track a lot of endpoints that we know are pretty important in a much more specific way and capture a lot of human diversity that we couldn't do before kind of makes up for the lacks. And I will say, if you want to get a lot of press, teach your brain cells to play Pong, because man, that has driven more traffic than anything. The autism thing, that's cool, but the Pong. Yeah.
Thank you, Alexandra, for the fantastic talk. In recent work, it has been published about the relation between plastic contaminated with bisphenol A and the development of cancer, specifically breast cancer. Can you comment about this?
Yeah, I always feel like, oh, I'm done with bisphenol A. So the problem with bisphenol A is that it does bind to the estrogen receptor, but it binds so weakly that I don't think that's the mechanism. And the one paper that I contributed to that 10,800 was pointing out that both the binding was so small that when you think of it in the context of both your endogenous estrogen as well as the phytoestrogen that you're exposed to, the chances that that molecule is going to bind to an estrogen receptor is pretty small. So I went in and transcriptomically, it looks like what it does is it just actually, I think I can describe it two ways. One is it made the transcriptome more unstable. We had a lot more or poorly correlated genes, but it also appeared to hit the estrogen-related receptor gamma, which is, and it binds, we know from protein crystallization data that it binds very, very tightly to that. We don't know a lot about what the estrogen-related receptor gamma does. So it is quite possible that long-term exposure to bisphenol A is causing a small increase. I will say one of the things they did is when they replaced bisphenol A, they replaced it with bisphenol S. And guess what? Bisphenol S binds even better to the estrogen receptor gamma. They designed it to not bind to the estrogen receptor, not thinking that there was a whole nother receptor they needed to think about. So that was one of the reasons why I tell people you really need to know molecular mechanism, or you might just be digging the hole deeper. So do I think it's a contributor to cancer? There have been no definitive studies because they're so hard to do, because the people who are exposed to BPA tend to also be people who live in cities, people who are living, the urban lifestyle that is cancer causing. Do I think the potential mechanisms are there? Absolutely. I don't worry a lot about BPA, but all else being equal, I would prefer if we designed it out of our plastics. So I think if we had, 3 million breast cancer patients and we had their lifetime BPA exposure, yeah, I probably think we would see something small there.
I have another question about the aryl receptor.
About what?
The aryl receptors. You mentioned about the aryl receptor.
Oh, the aryl hydrocarbon receptor. Yes.
Do you think it's a kind of important transcription factor to respond to pollution?
Yes. So we were sort of stuck with that because there's only one significant transcription factor that was in that entire list, but it was common to both, and it mediates your response to dioxin. So we know it is very important. It's also very important developmentally. And I think dioxin is one of the few chemicals that we know is an obesogen. That's kind of one of the ones where we're pretty clear about that because PPAR gamma and things like that. The problem we had was we had no way to really connect the exposures very well because we don't have a lot of transcriptomic data. And when we did like the ARNT pathway, we didn't see a lot. That may just be a limitation of the pathway. If I were running an epidemiology study to look at gene-environment interactions, I would definitely prioritize that. But in terms of candidates for chemicals, most people are not exposed to a lot of dioxin. I don't know that there's a lot that we could look at that would really be there. So we thought it was significant, but we kind of hit a dead end there. If it had been a CYP enzyme that metabolized, something we know about, or alcohol dehydrogenase, well, we know what those chemicals are doing or why you're having that effect. But ARNT, it could be anything, we don't know.
Meanwhile, you think your questions, I will read one from Zoom.
Sure.
And that's from Dr. Marta Vasquez, related to the question. She asks, do your studies consider different human groups, or are you expecting some correlations to be universal independently on what human groups you are studying?
So we are doing this with the data we have. We don't have the funding to do independent data. So we are literally just vacuuming up what is in the GWAS catalog, which is a mixture of studies. But those studies do kind of tend to skew towards European or American populations. So I won't say that they capture a lot of genetic diversity there. But we had to kind of start with the data we have, and I think one of the things we probably could do is we picked the same sort of unforgiving p-value cutoff that most GWAS studies get because their signal-to-noise ratio is not great. But if we're trying to look for gene-environment interactions, it's probably not a bad idea to lower that a little bit, and you could get some of the effects that are less frequent or harder to see, and that would help with the diversity issue. But one thing I will say is that one of the things that bothers me a lot about polygenic risk scores, you build that on a European population, that is not going to predict asthma in China. It just won't. It won't because the environment is different. It won't because the genetic background is different. So many things are different. So I do think that it's important to be quite cautious about the GWAS data and the limitations that we have right now. We have personally tried to do the best to kind of diversify and grab up as much as we could. But the data available is the data that we have.
¿Alguna otra pregunta?
Yeah, just continuing the last question. What would be your guess if you perform a GWAS to a mixed population of like Latin population? And yeah, what would be your guess with this asthma and diabetes correlation or overlap?
I think the overlap is just a feature of the fact that the human genome only has so many points of failure. And those points of failure, they may, so, kind of remember is that we are reducing this for each thing that we're looking at, so each gene, there's 5 or 6 SNPs. So we may be kind of biasing it towards SNPs that are common in, more common in a European or American population. But I suspect that if you had a broader genetic diversity, you would have more SNPs, you would definitely have a few more proteins, but I don't know that you would have 150 different proteins. I think that the genes that we kind of understand to be involved in diabetes are pretty well represented in this. But there are ways that we could kind of tackle that. So one of the things that, if you were looking at in a population and you cannot afford to, do whole genome, sequencing to get all the SNPs, we can kind of use our cholesterol molecular interaction network to say, hey, this is where you should look to do a little bit of dimensionality reduction. So when you think of those 90 genes that are implicated in cholesterol, only 3 of them had known SNPs that are implicated, 5 of them had known SNPs that were implicated in diabetes. I'm sure if you looked hard enough in broader populations, that would be a little bit larger. So I think that being able to tie the data that we have on the population that we have to potential environmental factors makes it easier to then look for gene environment interactions in broader populations, even if you have to be mindful of where the data came from.
Okay, thank you.
Another question? Well, I have one.
Mhm.
Regarding the SNP in the promoter region of the gene that you were describing,
Mhm.
so that will make it like if it increased expression
We don't know.
or like.
So this is one of the things, because at first we thought, well, the easy, obvious thing to do is to take our list of genes and look at EQTL studies. And then we can have, we won't have this hair-pulp network of interactions, we'll have directions, and that'll be such a cool paper. What we found out was, especially for polygenic diseases, but in GWAS in general, you tend to get hits in regions that are significantly upstream of the transcription factor binding site. Not a lot, not like 500, but 100. So we had to extend our search space a tiny bit. You also do get a lot that are in the introns. So one of the things I didn't mention, but I think it's kind of important to think about, was that both GWAS and diabetes were highly enriched for genes with known splice variants. And that's probably not an accident. It's probably also true of every polygenic disease you can think about. So, there's a limit to what we can do. So we abandoned EQTL for that reason. And we just decided that we would then focus on, we call them moonlighting genes, genes that have a lot of functions or a high semantic space. But it is specifically for that reason. You have some SNP that's sitting somewhere, a couple hundred base pairs away from where you think it would be doing something, and you do not know the effect at all. It's just.
Okay, so if there is no further question, thank you very much, Alexandra.
Okay, thank you, everyone.
Recording stopped.